Hi students, we just completed our first lecture exam, so well done everyone. I will have those graded and have your grades posted for you sometime by the middle of this week is my hope. So it usually goes pretty quickly. The multiple choice questions are self-graded, and then I just have to go through and grade all of your short answer questions. So I should be able to get that done before midweek. At least that's my goal. So we're going to go ahead and move on now, and our next big topic is this topic of inheritance, right? Or how we're actually passing on traits from one generation to the next. So all of the beginning content of this class was sort of building up our knowledge and our skill base so that we could then talk about inheritance and genetics, right? So we needed to understand what a cell is and what a cell does and how cells divide and how they replicate and how genes are turned into proteins. We needed to understand all of that information so that now we can get to sort of the meat of the class, the heart of the class, which is, all right, we know that a segment of DNA called a gene codes for a protein. And those proteins then are what exhibit in us as our traits, right? So they give us our physical traits. They give us our, what we're going to call metabolic traits, if you will, so the way that our bodies work. So all of this information is key so that now we can talk about modes of inheritance. And in this particular week, week 5 here, we'll be talking about topics that, again, are hopefully somewhat familiar to you all. Hopefully it's probably maybe been since high school biology, but maybe you remember some of these things, right? We talked about Gregor Mendel and the work that he did. We understand single gene modes of inheritance, dominance and recessives and phenotypes and genotypes, and we're going to cover all of that and more in this particular unit of information. So again, there's quite a lot here to go through, to be honest with you. So this is kind of another big week, just like our first week was a big week with lots of information. This week is another big week as well. So we've got sort of the standard set of assignments as we do every week. This week, you'll notice I do have 3 actual lectures for you all because, again, we have a lot that we need to cover this week. But again, it should be mostly review, I feel like. There might be some things that you've forgotten over time that are going to jog your memory, and you're going to think, oh yeah, I remember that. But hopefully nothing is too completely new for you. Assignments are the same ones we always have. I've already assigned the 3 adaptive practice modules, which, by the way, I am incredibly impressed by the fact that each week every student is doing all of these assignments. You're all turning them you're all turning them in, and that's fantastic. I rarely have that in a class, so kudos to all of you. I've also really enjoyed your discussions. You guys are doing a fantastic job of replying to the prompt and also really kind of critically thinking about the issues that we're raising and also responding and replying to each other. So again, I appreciate all of that very much. It makes grading these so much better when I have sort of those good quality responses and replies to read through. So thank you very much. And then the check-in week, the check-in survey for this week, which again, I will probably make it available on Wednesday, which is kind of what I've been doing as a standard. One other thing that I wanted to bring up and I wanted to mention is that you all received an email from me last week, and that email actually came from ThingLink. Okay, and I'm showing you an example. This is an account that I use as kind of my dummy student account so that I can see what the student side of things looks like. So I sent myself this email as well, and so you received this email that says, hey, please follow this link to create your profile on ThingLink. If you remember, ThingLink is what we are using to do this project. So under course materials, if you go to the media project, I have information on what we are doing. So I've got instructions for the project. I'm giving you a link to an example project, which I'm going to show you all in just a second. The images that you can choose, the guidelines, a little bit of information about how to do a peer review, and then some evaluation forms. It's not particularly difficult. It's not very hard, but the idea is I want you to take an image, and you get to pick from like 4 or 5 different images, and I want you to tag this image with information. So ThingLink, if you're not familiar with it, is just a different presentation tool. Okay, there's lots of presentation tools out there. I happen to like this one because we can take a static image and make it more dynamic and layer information in one single image versus a PowerPoint presentation or a slide presentation or something like that. So this is an example from a student from a couple of years ago, actually, and this is the image that she chose. So she chose an image looking at DNA technology or biotechnology, which we're going to talk about. And so she chose this image, and this is one of the stock images that you are able to choose as well. And then what ThingLink allows you to do is ThingLink allows you to tag the image. You add things to it to tell a story, right? So to give your audience some information. So if we click on these little blinking buttons, sort of things that are here that are blinking, there's different things that this is going to tell us about. So if I click on this little star right here, it's going to take me to a dictionary.com definition of what recombinant DNA is. So in this case, what we're doing is we're creating recombinant DNA, and she's going through the steps and the process by which we do that. She's also included a video talking about what DNA is. So this would link to a YouTube video about DNA. Some of the other things she included definitions like what's a plasmid, what are sticky ends. She also has information on, again, how this process works and what it looks like. So she's added a tutorial here that you can actually go to and see. And then she put examples of things that you can do with these recombinant DNA. So again, this is what ThingLink does, right? So again, it's really simple. It's fairly easy to do. Once you are signed up and all ready to go, you will be able to hit this button that says create, right? It's giving you lots and lots and lots of different options here. The only one you really need to do, if you want to play around with these, be my guest, but the one that I'm recommending is just clicking on image. It's going to bring up this little, I don't know if it's going to now, but previously it brought up a video of you know, how to tag an image and things like that. So it takes you through the steps and it takes you through the process. And again, it's pretty simple to do. And unfortunately, actually, I'm not sure why it's not actually working right now. That's slightly frustrating. Let me try that again. It might be because I'm in the process of recording, and so it's not going to show me the other end. But in any case, you click image here, and then again, you'll have an image, and there will be a toolbar, and the toolbar will let you do things like, oh, would you like to add a link here? Would you like to add a definition here? Would you like to add another picture here? Very simple, very easy to do. What I'll do then is I will take all of the projects, and I'm going to put them in a folder called Student Projects. And this is so that we can share these all out for the peer review process that you all are going to be a part of. So again, simple. This won't take you long. It just takes a little bit of pre-planning in the sense that you need to kind of choose what image you want. I give you different scenarios on how to frame your presentation. So thinking about audience, like who are you presenting to? Are you presenting to a biotech company? Are you presenting to a bunch of high schoolers learning about genetics? Are you presenting to a community health clinic organization? So again, I give you some sort of framework to work within in order to put these presentations together, but they're pretty, I think the instructions are fairly clear. If, of course, you have questions as you go along, please let me know. But once again, you have to start with this email and accept my invitation so that you can be part of my class, because I won't be able to view your project if you're not part of my particular class. Okay? So I think that's it for this week. Nothing else that I can think of. Once again, if you all have, I'll go back here to course materials, learning modules. How about we do that? If you all have any questions, please let me know. And again, I'll have some grades for you soon on your exams. I'm sure you all did fantastic. And then also I'll follow up with everybody on my Wednesday weekly email. So take care, everybody.